I'm Bronwyn Pinsly. I'm from Westfield State, Massachusetts. I am a senior studying biotechnology. The topics that the lab is researching involve organoids. Organoids, in simple terms, are essentially just miniature brains made from stem cells. We use these organoids as models for researching neurodevelopment. The lab has many ongoing projects, one of which is the aerialization project, which looks into how the cortex develops. Along with that, we're also engineering hydrogels in order to make optimal organoids. These hydrogels will secrete supplements to the organoids in order to create either posterior or anterior organoids. The things I'm most interested in terms of the project are just expanding again the knowledge of the cortex and neurodevelopment, along with learning new bioengineering and biotechnology techniques. So yeah, that's what I'm most interested about.
My name is Bella. I'm a graduate student. I'm a third-year student in the Department of Anatomy and Neurobiology in the Watanabe lab in the School of Medicine. So broadly speaking, the mission of the lab is to create human tissue-based system for modeling the brain. So our lab uses human stem cells to create brain organoids, specifically cortical brain organoids. So we use human embryonic stem cells or induced pluripotent stem cells to create brain organoids, and that way we can use these as a platform to study neurodevelopment, neurodevelopmental pathways, different signaling pathways and molecules, and how they work in the brain, and how they create patterning in the brain and development, and how the different regions of the cortex are developed versus anterior versus posterior regions of the cortex. And with these brain organoids, we can also study neurodevelopmental diseases such as Fragile X syndrome, which we are currently working on in the lab. But we overall want to use these brain organoids as a really powerful tool to study human-specific features of neurodevelopment. So having Bronwyn in the lab has been really great. Our lab is really small. I'm the only grad student, so it's really awesome to be able to collaborate with other young students and kind of introduce them into the world of brain organoids and stem cell culture, especially because I know Bronwyn is really excited about bioengineering, and our hydrogel project is a perfect fit for him. So I'm so happy that he has the chance to interact with that project and get to know what goes into creating a project like this and also seeing it through to the end as we're nearing publication. So I'm really happy that he can be here, and I'm just excited that I get the chance to be his mentor because he's really awesome.